Hello, biologists. I wanted to talk to you guys about the first step of protein synthesis known as transcription. So today I hope that you guys will be able to describe how DNA and RNA both play a role in the making of a protein. And I want you guys to understand how this process occurs, transcription. How do we make this molecule of RNA using what we know about the DNA? So remember, in central dogma, it's a two-step process. This first step process that we're focusing on now is called transcription, and it occurs in the nucleus. And it involves using the sequence that's stored in our DNA strand in order to make a complementary sequence or molecule of messenger RNA. In this whole process of making a protein or protein synthesis, we see that there are several different types of RNA molecules involved. In the first step of protein synthesis, we see that we make messenger RNA. So we're going to be talking mostly about messenger RNA this week, but know later on when we're out in the cytoplasm using this messenger RNA, in order to convert this information into a functional protein, we'll also need the help of ribosomal RNA and transfer RNA, which we'll talk more about later. So while transcription is occurring, again, in the nucleus, we need to figure out how to take this sequence that's stored in our DNA and preserve it in this molecule of messenger RNA so we can send that message out into the cytoplasm for protein synthesis to be completed. So know that this messenger RNA, this molecule we're making, is a copy of just a portion of the DNA strand. Remember, a lot of the DNA that's stored in the nucleus is non-coding, so it's only just a segment of the DNA known as a gene that contains the information that we really need to use to make a functional protein product. So after it's made in the nucleus, again, it's going to leave through a nuclear pore out into the cytoplasm where the rest of protein synthesis, known as translation, can occur. So let's talk about how these molecules of messenger RNA are made in the nucleus. So here we're looking at a segment of DNA, and this segment is going to be copied with the help of an enzyme known as RNA polymerase. And this enzyme basically reads the segment of DNA and pulls in the necessary nucleotides that would base pair with those nitrogenous bases. So we would read this from left to right. We're going to start with our first nitrogenous base of our DNA molecule, and we would think what pairs with cytosine, C. And this enzyme would then pull whichever one of these nitrogenous bases is needed to start building that messenger RNA. So we would have the enzyme first pull over a guanine because we know G pairs with C. And this enzyme would continue reading, pulling in the necessary nucleotides. So here, guanine, we know pairs with cytosine, so a cytosine would be pulled in. And this is the beginning parts of our messenger RNA molecule that we're building. We know T pairs with A, G pairs with C, and so on. When we're looking for a base pair for adenine here, remember this is our molecule of DNA, and we're building a molecule of RNA. Remember, in RNA, we did not see any thymines contained in that structure. Instead of thymines, we're going to see that uracils or U's are going to be pulled in to base pair with adenine when we're building our molecule of RNA, because RNA does not contain any thymines. The base pairing continues, and we would finish building our messenger RNA product, and eventually these nucleotides would all be bonded together, and this single strand that we see down here at the bottom would leave the nucleus, go out the nuclear pore, and then finish protein synthesis in the cytoplasm. So that's an overview of this first step called transcription. Again, that it occurs in the nucleus, and the product, known as a molecule of messenger RNA, then leaves the nucleus, and the second step, translation, is going to be finished in the cytoplasm.